Well, welcome everybody. I'm Michael Malin from the School of Immunology and Microbial Sciences and the Faculty of Life Sciences and Medicine. And on behalf of King's Health Partners and King's College London, I'm delighted to welcome all of you to this evening, as well as our guest, Professor Jennifer Doudna, to this evening's Rosalind Franklin Lecture. This is one of the prominent events in the year's King's International Lecture Series. But before proceeding to the main business of the evening, I just want to provide you all with a little bit of context to this lecture series. It celebrates our Academic Health Sciences Centre, King's Health Partners, which was first designated by the Department of Health in 2009, and in 2014 was accredited for a further 5 years. This partnership is the practical and philosophical integration of King's College London with the 3 South London NHS Foundation Trusts, Guy's and St Thomas's, King's College Hospital, and the South London and Maudsley, with the primary aspiration of trying to integrate excellence and innovation in research, education, and patient care, locally as well as internationally. In terms of research, King's Health Partners seeks to support the alignment of the very best basic discovery-orientated science with more applied translational and clinical research. Now, hosting the Rosalind Franklin lectures is always a great privilege. In particular, these exemplify the central importance of fundamental research to understanding and improving human health. And as you will know, these lectures are named in recognition of the life and achievements of the King's scientist, Dr. Rosalind Franklin. Dr. Franklin, a biophysicist and crystallographer, is arguably one of the true giants of biological research. She is best known for her groundbreaking structural studies on DNA, which led to the elucidation of the DNA double helix. The enduring impact of her work continues to resonate to this day, one illustration being the growth of genomic medicine based on DNA sequence in individual patients, and another would in fact be the work that you're going to hear about shortly. We've been fortunate in previous seminars in this series to have heard some truly outstanding scientists, including Randy Sheckman, who discussed his work on protein secretion autophagy, Stefan Hell, who described the development of super-resolution fluorescence microscopy and its applications, and Roger Chen, who described his pioneering work on the exploitation of fluorescent proteins to guide surgery. Tonight, we are really privileged to welcome Dr. Jennifer Doudna from the University of California, Berkeley. As our host, Professor Franca Fraternelli, will outline in greater detail, Jennifer is a chemist and structural biologist by training, having worked on topics such as catalytic RNAs and RNA silencing. But around 10 years ago, she got interested in a then mysterious pathway of bacterial resistance or immunity to phage infection called CRISPR. Among a number of projects, and together with her collaborator Emmanuel Charpentier, Jennifer set about engineering CRISPR machinery to repurpose it as a tool for the targeted alteration of genomic DNA. Their remarkable success in this endeavor was reported in June 2012 in what is truly a landmark publication. This has opened the floodgates for a multitude of applications, some of which Professor Doudna will discuss today. Jennifer is currently a professor in the Department of Molecular and Cell Biology at the University of California, Berkeley, and an investigator with the Howard Hughes Medical Institute. So without further ado, I'm going to hand over to Franca from the School of Basic and Biomedical and Medical Biosciences, who will introduce the speaker. Franca, thank you very much for hosting today's seminar.
Thank you, Mike. And I have to say that when we decided to invite Jennifer Doudna back in 2015, we knew she had a busy life, an interesting life, but we had not anticipated how much this was until she kindly, enthusiastically accepted, but replied that she could make it in October 2017. So now we know how fast the ripples of her discovery have permeated many scientific fields, including cellular biology, biochemistry, genetics, and medicine. The career path of Jennifer epitomizes the importance of being grounded in basic discipline in order to develop rigorous and quantitative tools. She has been able to bridge two related and complementary disciplines to be open-minded and ready to collaborate across fields, to be forward-looking, and to exploit the potential of her scientific discovery. She was an undergraduate in biochemistry at the Pomona College in California. She did her PhD in biological chemistry and molecular pharmacology at Harvard Medical School with Professor Schotzak. She was then a research fellow at the University of Colorado with Professor Tomaszek, and then became professor herself at Yale in the Department of Molecular Biophysics and Biochemistry. Since 2002, as Mike mentioned, she is professor at the University of California, Berkeley, in the Department of Molecular and Cell Biology and Department of Chemistry. So amongst the achievements that Mike mentioned, she is a member of the Academy of Sciences and National Academy of Medicine and the American Academy of Arts and Sciences. She's also a foreign member of the Royal Society. She had many honors and prizes, which I won't mention all today, but amongst them I selected some. So she was awarded the Breakthrough Prize in Life Science together with Emmanuelle Charpentier. She got the L'Oreal Prize for Women in Sciences. She was a recipient of the Heineken Prize for Biochemistry and Biophysics. And interestingly, she has been listed as very recently, as one of the 100 most influential people in the world, but probably more exceptionally, amongst the 100 leading global thinkers. She has in common with Rosalind Franklin passion and determination in her scientific approach, interest in molecule, nucleic acids, and structure biology. And while Rosalind was amongst the one who laid the basis for our understanding of DNA code and replication, Jennifer has laid the basis of DNA editing and manipulation. So without further ado, I leave lecture field to Jennifer, which will give a lecture on CRISPR system, Nature Toolkit for Genome Editing. Welcome.
Well, good afternoon. Thank you for that very generous introduction, and thank you for welcoming me to King's College. I've had a fantastic day today talking science and meeting with the students. That was just great. The first time that I think I ever read about Rosalind Franklin was when my father gave me a copy of the Double Helix when I was about maybe 12 or 13 years old. And I read that book, and it kind of blew my mind. I was amazed to think that scientists could actually come up with ways of doing experiments that would reveal the structure of a molecule. It was kind of mind-blowing. And I think for me, that was probably the moment in my life when I first started to imagine that I might want to become a scientist. So it was, of course, very exciting later to learn more about Rosalind Franklin and her legacy. I can tell you I never would have imagined that I would be standing here and giving a lecture in her honor, and that's really very special to me. So what you didn't hear about in the introduction is that, and it's especially for the students here, I grew up in a small town in Hawaii. Nobody in my family was a scientist, and the only reason I'm here is because I always loved math and science when I was growing up, and I just tried to never let anybody tell me I couldn't do that, do what I really wanted to do. And I've tried to maintain sort of that approach to science of just trying to maintain a little bit of a childlike curiosity about nature and the questions that I've been interested in addressing over the years. So the story that I'm going to tell you today is really, I think it's a real example of curiosity-driven research going in a very unexpected direction, because I certainly didn't start out working on CRISPR biology thinking that it would go the way that it has. So with that short intro, I'd like to just start off by pointing out that, and this is hardly a slide that I need to show here, but you know that that the DNA in a cell is really the information, sort of the chemical information that tells cells how to grow and divide and become a tissue or an organism. And I think really it was the discovery of the structure of DNA that was done here that really kind of sparked the era of modern biology. It started to inspire scientists to think about how you could understand all of this information that was encoded in DNA. Could you ever really know what it meant? And could you eventually manipulate that information? Could you copy it? Could you sequence it? Could you cut it? And eventually started thinking about what if you could actually alter it in cells? What if you could make changes to the DNA code in cells in a precise fashion? And this is an idea that's really been around for decades. I can remember when I was a student, graduate student in the 1980s, reading about the work of people like Peter Dervin, a chemist at Caltech, whose work showed that you could use DNA oligonucleotides to recognize sites in chromosomes, and that was one of the first ways that people started to map genes back before there was long before there was a human genome sequence that was known. So this has been an area of science that's been very active for a long time. But I wasn't involved in it, right? I was doing other things. And so I actually got into, when I first got into science, I was sort of, we were taught the central dogma of molecular biology about, how DNA encodes the information that's required to make an organism, make a cell, make an organism, and that information ultimately is basically expressed in cells in the form of proteins. Let's see, I got to make sure I get the right, here we go, maybe. In the form of proteins that, we were taught do all the interesting things in cells. And then in the middle was this molecule called RNA that is, a transient copy of some of the genetic information. And if it's a messenger RNA, then it's a molecule that encodes proteins. And so, when I was first learning biology, it sort of seemed like a pretty boring molecule, not something you wanted to spend much time on. And so I was really excited when I got to graduate school, and I learned about examples of RNA molecules that do really interesting things in their own right. They don't encode proteins, but they actually function at the level of RNA to do things in cells like even catalyze chemical reactions if they're a ribozyme. And so that got me wondering about the structures of those RNAs. What might they look like? Did they look like proteins, or did they look like something else? And so that set me off on a path of doing, when I first started doing X-ray crystallography, was thinking about how do we solve the structures of RNA molecules that are functional in biology. And in a really kind of an interesting and very circuitous way, that kind of led me to CRISPRs, because when I started my faculty career, I started in the mid-90s at Yale University, and I was working on the structures of ribozymes at that time. And then about 8 years later, I got wooed away to my current university, University of California, Berkeley. And one of my motivations for moving to Berkeley was that I was very excited about the opportunities to take what we were learning about the structures of RNA, and we were doing a lot of biochemical experiments as well, purifying molecules and testing their activities. And I thought it would be really fascinating to take that kind of detailed molecular knowledge and be able to query it in cellular systems. I wanted to really start doing more sort of cell-based experiments. And so when I moved to Berkeley, at that time, it was early 2000s, a lot of the buzz in the RNA world was actually not about big molecules like the ones I'd been studying, but about very small molecules of RNA called microRNAs, so RNAs that are involved in controlling gene expression in eukaryotic cells. And so we were excited to get started working on some of the molecular mechanisms that allow small RNAs, like microRNAs, to be made in cells, and the enzymes that are involved in processing those RNAs. And that led to a sort of a phone call that I got one day in my office at Berkeley from a colleague of mine, Jill Banfield. And Jill is a scientist who works on metagenomic sequencing. So she goes to interesting environmental sites, collects samples of organisms that are growing there, and then sequences all the DNA to figure out what kinds of bacteria and other kinds of microbes might be existing in these environments, and also the kinds of phage that they might, or viruses they might be interacting with. And that led to what seemed like a very, kind of just a curious observation. Nobody knew what it meant at the time, but she had, I think, a kind of a premonition, or just an inkling that it might involve RNA. And her observation was that in the chromosomes of many bacteria, and this is true for about half of the sequenced bacteria, there's at least one example of an array of sequences that have a very distinctive pattern, where you have a set of repeated elements that are about 30 to 40 base pairs long. They often have palindromic character to them, so they can have sequences running in opposite directions, so they could potentially form hairpin type self-structures. And then in between these repetitive units were a series of unique sequences. And initially, it wasn't clear what these were, but they were always different. In every organism, they were different, and every one of these spacer sequences was different from the other. And so these had been called CRISPRs, and I think it was actually Francisco Mojica that coined that acronym. But nobody knew what they did. And the reason Jill called me was that in the middle of the 2000s, 3 publications from bioinformatics labs pointed out that in many cases you could map these intervening sequences to sequences that were found in the viruses that infect these bacteria. So it was a really interesting hint that what these arrays, these CRISPR arrays, might represent was some kind of an adaptive immune system in bacteria, some way that bacteria could store the information from viral infection, store it over time in the genome, in this array, and then somehow deploy that information to protect cells from future infection. The question was how? How would they do that? How would that work? And Jill's idea, and a few other people like Eugene Koonin at the time were thinking about this, was that these sequences might actually be operating at the level of RNA. So she literally Googled, who at Berkeley works on RNA, and my name popped up. And so she called me and said, I think you need to come and talk to me because I have something very interesting to tell you. So that was how I heard about CRISPRs. And the cool thing about these is that they don't exist alone, but they're actually found in the genome next to CRISPR-associated, or Cas genes. So these were genes that, again, bioinformatically, always seem to co-vary with these CRISPR arrays. And the mystery was, what do they do? It sort of looked like something that was clearly being conserved in bacteria, but nobody had done an experiment at that point to figure out how they worked. And so that really launched us on a fascinating journey. So we started working on this system a couple of years, within a couple of years of that first discussion with Jill. And as I'll tell you, we started doing fundamental biochemical experiments and structural biology to understand the molecular mechanisms at work in these systems that are in fact adaptive immune systems. And so today, what I'm going to do is I really want to tell you a little bit about how we got into this experimentally and how it led to realizing that these CRISPR systems could be harnessed for gene editing. And then I want to say a little bit about some of the work that we're doing now to apply these gene editing tools in systems that I think are going to be very important both for fundamental understanding but also for potentially in the future clinical use in people. And then at the end of the talk, I want to say a little bit about ethics, because this is also part of my journey, is realizing in the course of this work that research that we were doing in the lab that we were sort of very narrowly focused on was actually going to likely to have societal impacts that went far beyond what I had ever imagined, and realizing that I really needed to get involved in that discussion and educating people about this technology, what it means for the future, and how we can ensure that it's used in a responsible way. But let me start off with talking about a little more about CRISPRs, and I thought I would just show you a cartoon that illustrates an overall idea of how these CRISPR pathways work. And I should say that there are many varieties of these pathways in nature. But overall, the way they operate is they allow cells to detect foreign DNA that gets into the cell. And this is an example. Here we have the edge of a bacterial cell that's being infected by a virus. And when this virus infects, the DNA from the virus is injected. And if this lucky cell has a CRISPR array, then it has a mechanism of detecting this DNA and stealing a little piece of it and integrating it into the CRISPR array. And this doesn't happen randomly, but the piece gets inserted on one end of the array, and the integration involves copying of the repeat so that you maintain the structure of the array. And that's actually really important for the way this works, because what the cell does after that is to transcribe the array into a precursor CRISPR RNA, and you can see here the result of these palindromic sequences in the repeats gives rise to RNAs that have the ability to fold back into these hairpin type structures. And that's required in many of these systems for processing of the RNA into the functional units. So each resulting RNA has a sequence that comes from a virus originally, and then sequences on either end that are the same in every RNA. So it's a nice way of the cell sort of marking this RNA as a CRISPR RNA. And these RNAs then assemble with one or more of the Cas proteins to form complexes of protein and RNA that search through the cell looking for sequences that match the sequence in the CRISPR RNA. And when that match is found, base pairing allows a tight association of the complex, and the CRISPR, the Cas enzymes are able to cut the foreign DNA. So, and this, was all worked out initially by some very beautiful phage genetics that was done by folks at various places, but interestingly, including Danisco, a yogurt company. So they were working on this to figure out how to protect their yogurt cultures from phage infection. And then subsequently by some various kinds of biochemical studies that were done to figure out how the components of this system are operating. And so I want to focus today on this step over here, sort of this interference phase where these complexes are able to search out and ultimately destroy foreign nucleic acids. But I just want to point out that there's a lot of really interesting biology in these steps of the CRISPR systems as well. And we in my lab and others have been working away on these too. And I think it's not unreasonable to think that we'll find principles of these pathways that are applicable both to understanding the fundamental biology, but also potentially useful as technology kinds of tools in the future. So one of the things, as I mentioned, one of the things that's really interesting about CRISPR systems is that there's many flavors of these in biology, many types. And roughly we can break them down into two overall classes, class 1 and class 2. The class 1 CRISPR pathways all feature multiple enzymes, multiple proteins that assemble into these effector complexes with the CRISPR RNA. So these are just showing you individual genes that encode proteins required for protection of cells from phage infection if they have one of the class 1 CRISPR pathways. The class 2 systems are different because in each case they have a single gene. It's typically encoding a pretty large protein that is required for protection. And so in some ways you could say, well, maybe it's sort of a simplified system, but it's not quite that easy to make that connection because these proteins are really different from each other, and they're also very distinct from anything encoded by the genes up here. So it's not entirely clear what the evolutionary path of these systems is, except that they're clearly diverging very quickly, probably to respond to the ways that phage are able to fight back against CRISPR pathways. So when we first started working on CRISPR systems, my lab was actually focused on, we were working on these types up here that have multiple proteins. And they were really fun to work on, as a biochemist and a structural biologist, because these proteins assemble on CRISPR RNAs and they form these beautiful structures. We could look at these by crystallography and by cryo-electron microscopy. And you could make mutations in the proteins, and they had affected their ability to to protect cells from phage. So it was really nice to be able to go back and forth between doing pretty hardcore biochemistry and biophysics and then doing cell-based assays and testing mutants in bacteria and things like that. So it was really nice to go back and forth that way. And then I went to a conference in 2011, and I met Emmanuelle Charpentier, who is a medical microbiologist. At the time, she was working in Sweden. And she told me about research that her lab was doing on one of the class 2 CRISPR systems. And this was a system that she had come to in her research on an organism called Streptococcus pyogenes. It's a bacterium that causes the flesh-eating disease in immunocompromised patients. And in the course of her work, she had come across the CRISPR system in that organism and its potential role in pathogenesis. So she'd gotten very interested in the biology of the CRISPR pathway. And what was interesting about the CRISPR system in Streptococcus pyogenes is that it belonged to this class 2 family. So it had a single gene encoding a large protein called Cas9 that was genetically implicated in protecting cells from viral infection. But at the time, nobody knew what the function of this protein was. And so when we were walking around the streets of old San Juan, Puerto Rico at that meeting, we started talking about the possibility of working together to figure out the function of this protein. And so that really launched us on a fabulous collaboration, an international partnership where we had students in Emmanuelle's lab, a postdoc, and an undergraduate student in my lab who teamed up to work on this. And initially, these students had never met each other, so thank goodness for Skype. So they were communicating a lot over Skype. And I've told this story before, but another sort of curious thing that was just one of these serendipities of science was that in this collaboration, the person in my lab doing the research, Martin Žinek, had grown up in the Czech Republic. And the student in Emmanuelle's lab doing the work on her side of the Atlantic was Krzysztof Czajlinski, a guy from Poland. And these two people, when they started talking over Skype, they quickly realized they had grown up on opposite sides of the Polish border, very close towns. And they both spoke the same dialect of Polish. And so who could have predicted, right? But it was a really interesting cultural connection they had. And that was probably no small ingredient in sparking the chemistry of this collaboration. So they got to work, and they quickly figured out that Cas9 was a really pretty cool protein. So it turned out that this protein is able to cut double-stranded DNA in an RNA-targeted fashion, something that we had sort of hypothesized based on what was known about CRISPR systems at the time. This is a cartoon that shows a double-stranded DNA molecule binding to the Cas9 protein, this blue blob, with a CRISPR RNA guiding the binding site. So there are 20 nucleotides here in the CRISPR RNA, and remember that would be coming, if it's in a bacterium, it might be coming from a virus that had previously infected the cell. And this RNA forms a 20 base pair interaction with one strand of the DNA. So DNA actually has to open up inside the protein to allow two separate active sites to cut each strand of the DNA. So it's a double-stranded DNA cleaver. Now, there were a few surprises. So one of the big surprises that came out in our biochemical work was that this protein absolutely requires not only the CRISPR RNA, but a second RNA molecule called tracer, which is this red molecule right here. Now, this was an RNA that Emmanuelle's lab had originally discovered to be required for generating the mature CRISPR transcripts, the smaller units of RNA that are actually functional in the effector complex. But we found out that this RNA, rather than going away after that processing step, it actually sticks around because it forms a structure with the end of the CRISPR RNA here that is a handle for binding by the Cas9 protein. So you need to have both RNA molecules to assemble this effector complex. And Martin and Krzysztof also figured out that this targeting mechanism requires that the site in the DNA be next door to a little motif that's known as the PAM sequence. And for this enzyme, it's 2 GC base pairs, so it's very small, but it's absolutely required for DNA targeting. But by following those rules, having these 2 RNAs present and choosing sites in DNA that were adjacent to this GG PAM motif, we could get this protein to cut any desired DNA sequence. So, you know, I'm sort of thinking back to my graduate days, and we used to, you know, this was before we had everything on the internet, so, you know, we used to have our New England Biolabs catalog in the lab, and, you know, basically anytime you sat down to do an experiment, you'd run to the New England Biolabs catalog and look up, okay, what restriction enzymes do I need to use to cut my DNA, if you're doing cloning or something. And I sort of realized, wow, that's like taking that entire catalog and just cramming it into one protein, right? And you know, I've got this programmable protein that has evolved in bacteria to have this very flexible targeting capability. So it seemed really cool. And then even cooler was Martin Ginnek going back and sort of trimming away at the essential sequences in these natural RNAs to the point where he realized that these RNAs could probably be linked together. And so he ended up generating these single guide forms of the RNA that had a linker connecting the ends of the CRISPR RNA and the tracer RNA. Again, allowing this handle structure to form, but now requiring just a single transcript to be made. So you could program Cas9 to target different DNA sequences by simply changing the 20 nucleotides on the end of this RNA that otherwise had the same exact sequence down at this other end. And I think for us, you know, when Martin did that initial experiment, I think that was kind of the moment, kind of those proverbial moment for us when this project went from a curiosity-driven research effort to recognizing that this could be a very exciting technology because of the simplicity of being able to reprogram a single enzyme to cut DNA at a desired site by simply ordering or synthesizing a single fairly small RNA transcript. So, and I'll say a little bit more about that shortly, but we've been really interested in understanding fundamentally how this works. And one of the things that's really intriguing about this enzyme is that it literally opens up double-stranded DNA. So it displaces the base pairs that are forming in this targeted region of the double helix and instead sort of catalyzes formation of a RNA-DNA hybrid that turns out to be essential to the targeting mechanism. So until that hybridization occurs, cleavage of the DNA can't happen. So, and I'll show you briefly some data shortly that show that it's really quite a sequential process. And furthermore, the curious thing about this, or to me is still something we're still puzzling about and trying to figure out what's going on, is that this enzyme doesn't have an external energy source. Okay, so somehow it has to unwind DNA without hydrolyzing ATP or GTP. And it does so clearly very, very efficiently and very quickly. And so I'm not going to answer that question today because we don't really fully know how it works. But I think we've gotten some clues over the last few years. And one of the hints about how Cas9 might operate is the fact that it undergoes a very dramatic rearrangement in its protein structure when it interacts with nucleic acid. So I'm going to show you a little movie that morphs between different structural states of Cas9. And these are based on actual crystallographic structures that have been solved by several different labs. And the animation was made by an undergraduate student, Ben LaFrance. And so this animation starts off with Cas9 protein by itself. And as it binds to its guide RNA, you saw a big rearrangement in the structure of the protein right here that accommodates the orange RNA and in fact opens up a channel in the center of the protein where the guide RNA strand is located. And then when DNA binds, there's an additional structural change to accommodate the RNA-DNA hybrid that forms inside the enzyme. And then there's an additional rotation that happens in this part of the protein. This is a domain of the protein called HNH. It's one of the catalytic domains. It's the part of the enzyme that actually cuts the targeted strand of the DNA that's base paired with the RNA here. And that final conformational change doesn't happen right away. And in fact, all of the early crystallographic structures had trapped the enzyme in what was clearly an inactive state because that H and H domain was in the wrong place to cut the DNA. So we sort of, we knew that there had to be an additional conformational change happening. And that motivated a former graduate student, Sam Sternberg, to set up experiments to monitor what we inferred must be these conformational changes that happen in the enzyme in solution when it binds to RNA and then eventually to a DNA substrate. And so what he did was to engineer into Cas9 pairs of dye molecules that would allow us to monitor these conformational changes by fluorescence resonance energy transfer, or FRET. And so we knew that there were sort of the possibility for 3 primary motions in the protein. We figured out that each of those could be monitored using dye pairs that were placed on different positions on the enzyme to be able to detect these by FRET. And that led to a series of experiments that we've done more recently. So those initial assays by Sam Sternberg were done using bulk FRET studies. So we have a mixture of sort of a pot of molecules, and you're looking at behavior of the whole population at once. We thought, wouldn't it be great if you could actually do those experiments, but do them on individual molecules, a single molecule FRET experiment where you could monitor the behavior of molecules and then get much more precise information about these conformational states. And so more recently, we've been working with 2 labs, Ahmet Yildiz, who's a biophysicist at UC Berkeley, and on the other end of the spectrum, Keith Jung, who is an MD who works at Harvard Mass General Hospital and has been one of the leaders in sort of harnessing Cas9 for gene editing experiments, to do experiments in which we asked very detailed biophysical questions about the conformational state of the enzyme as a function of binding to DNA molecules that have different degrees of complementarity to the guide RNA. The reason being we really want to understand what allows this protein to not only to unwind DNA, but to really be pretty good at detecting on versus off-target DNA sequences. And furthermore, once we understand how that works in the native enzyme, could we make it even better? And so I'm just going to summarize some of the recent work we've done on this. So this is an example of how we can use these fluorescent placement of these pairs of dyes. So here you can see the structure of Cas9, and when it's in the inactive state, this active site domain is rotated over here. And so if we put a dye molecule in this position and a second one over here, these initially start off very far apart so we don't get much FRET. And when this domain rotates into position so it can cut DNA, now the dyes are quite close together and we get a very robust FRET signal. So it's a great way to monitor the conformational change that's associated with activation of the enzyme. And so I'm just going to show you very briefly what some of these data look like. And this is now work that was done by 2 students who teamed up, Yavuz Dagdas in Ahmet's lab and Janice Chen in my lab, 2 graduate students. And one of the ways they set up their experiments is shown right here, where we can tether the Cas9 guide RNA complex to a slide using a DNA oligo, and then flow in different DNA substrates that have different degrees of complementarity in the target sequence here to the sequence in the CRISPR RNA. And here's the PAM motif. So again, that's required for targeting. And so when we do these types of experiments, we could get a lot more detail about the conformational changes that happen than we got initially from these bulk FRET experiments. And so I'm just going to show you one example of these data. And so this is showing a comparison of FRET signals that we get for molecules that are binding to a perfectly matched DNA target substrate to DNA molecules that have different numbers of mismatches to the CRISPR RNA. And then over here, a molecule that has just the CRISPR RNA. It doesn't have a target DNA substrate associated with it. And so these are single molecule experiments. So we're looking at individual FRET signals from individual molecules, and then we're binning them. So that's what you're seeing plotted here. And where to stand? Maybe I'll stand over here. And so what you can see is that when we have a perfectly matched DNA target, virtually all of the molecules are in this high fret state. So remember, that's when the dyes are close together, right? So we know this is it's in an active conformation that's cutting the DNA, versus what we see for no DNA bound at all. So most of the molecules are hanging out in this low fret state, so they're in the off position, inactive state over here. And what was really kind of interesting was noticing that when we have molecules bound to DNAs that have different numbers of mismatches with the CRISPR RNA, we found that we started to populate this intermediate state here. It's not fully off, but it's also definitely not in this high fret on position either. So we've got a lot of molecules that are populating this intermediate, and that led to the idea that this really is there's sort of a defined mechanism by which this enzyme swings into action by going through these conformational changes that are really a function of not only binding to the guide RNA, but also to assembling with a DNA molecule. And it matters how much base pairing complementarity there is to the guide RNA. It actually affects the conformational state of the enzyme. And I'm not going to show you the data today, but we just published a paper very recently in which we were able to use that information. And again, this is in collaboration with Keith Jung's lab to design enzymes that are even better, sort of more accurate than the natural system. We figured out where the sensor is in the enzyme, and we could actually make mutations there that allow the enzyme to be more sensitive to even a single mismatch between the target DNA and the guide RNA than we find in nature. And so that sort of gives us hope that in the future, as we go forward understanding the mechanism, we can really use that knowledge to impact what's happening with this and the use of this system as a technology. But I want to say a little bit about genome editing. So why, you might be wondering, probably not, but you might be wondering, what triggered us to start thinking about this DNA cutting enzyme in the context of gene editing. And it really was because of all the beautiful work going on in many labs over the years to understand how cells repair broken DNA, and eventually how you could actually harness that to make very precise changes to genomes. And fundamentally, the idea is that in eukaryotic cells, when a double-stranded break occurs, cells have ways of detecting the break and repairing it by probably multiple different pathways, but two primary pathways are shown here. One is non-homologous end joining that leads to occasionally the disruption of the DNA sequence right at the site of repair, or by recombination, in which a template DNA with some base pairing complementarity to the site that's been broken can be recombined to integrate a new piece of DNA at the site of the break. And so by taking advantage of those systems, it had been possible for labs to engineer enzymes like zinc finger nucleases and talon proteins to recognize particular DNA sequences in a genome and make a cut, and then hand it off to the repair machinery to fix. And very powerful technologies, very exciting. Lots of people thinking about how you could use this tool. The challenge was that it involved significant protein engineering. So for every experiment, a new protein had to be engineered. So it's kind of back to the New England Biolabs catalog, right? You got to have a new enzyme for every single site that you want to cut. And the really cool thing about Cas9 is that nature had already come up with a way to program a single enzyme to cut many different DNA sequences by simply altering the guide RNA that it binds to. And so that was really the idea. Once we understood how Cas9 was functioning, it was pretty clear that this could be harnessed for a very different purpose, namely to make precise changes to genomes. So let me show you a quick video here that just illustrates how we imagine this might work in the context of a eukaryotic cell. So in a eukaryotic cell, unlike in bacteria, the DNA is inside the nucleus, and not only that, it's packaged in chromatin. So you can see the green histone proteins, these nucleosomes, DNA is wrapping around them. And somehow this bacterial enzyme has to search through the DNA in the nucleus to find a single site that matches the sequence of the guide RNA. When that match occurs, it opens up the DNA, forms this RNA-DNA hybrid, and then makes a double-stranded break that gets passed off to repair enzymes in the cell and allows sequences to be inserted or to be altered at exactly that place. So when we published our work with Emmanuel in the summer of 2012, we proposed this as a genome editing tool. And it was literally before the end of that year that several labs were already doing experiments to test this, including us, and figuring out that this was actually going to be a very widely applicable tool, basically working in many different types of cells and even in whole organisms. Keith Jung's lab doing the first experiments in zebrafish, showing that you could modify them at precise places using this approach. So, we've done a lot of work in the lab on sort of the mechanisms of Cas9 and related enzymes, and I thought that instead of telling you about that, which I kind of would love to tell you about, but actually I realized when I was talking to Mike Malum about this lecture that one of the things that this lecture series really highlights is science that starts off from a very fundamental perspective, asking a basic question about something in biology, that then morphs into something that's much more applied. And there is about a third of my lab now that are working on very applied questions and things that we want to do using Cas9 as a gene editing tool. And so I thought I would just tell you about some work that we're doing and ways that we're thinking about this as a sort of in honor of Rosalind Franklin and some of the work that we think is going to be possible in the future based on this fundamental understanding of CRISPR pathways. And so I wanted to point out that when we think about applying gene editing, especially in the clinic, and by the way, I think, in my own opinion, I think the biggest probably global impact of gene editing in the near term is going to be in agriculture, right? Because it'll really impact the largest number of people quickly in sort of areas of agriculture. But I do think that the opportunities in the clinic are just incredibly exciting and tantalizing to think about. And when we think about that, there really are kind of, in my opinion, three major challenges that we have to address. One is delivery. How do we deliver these gene editing molecules into cells or tissues? It's one thing to do it in the laboratory, but it's a whole different ball of wax if you want to deliver gene editing molecules into particular tissues or into a whole organism. How do we think about that? Secondly, how do we control the way that DNA repair happens? That's clearly an integral part of the way this is working as a gene editing tool. So being able to control the way that cells repair their breaks will be critical in the future. And then thinking about some of the ethical challenges that come up, especially when we think about using this in human embryos. And also questions like, how expensive would this be as a clinical treatment? Who gets access? Who pays for it? In my country, it's not clear. There's a lot of arguing over insurance and who's going to pay for something like this. So these are some of the things that we're thinking about. But today I'm just going to focus on this first point, which is delivery, and thinking about how we can sort of address that challenge. And of course, we're biochemists, so we're coming at this from what we know how to do, which is work with purified molecules. And so around the time that right after I published this work with Emmanuel in 2012, I got a call from Brett Stahl, who was at the time finishing a PhD at Stanford. And he actually wanted to come to the lab to work on Huntington's disease, which is one of the sort of very important type of neurological disorder that results from a gene that has a triplet repeat expansion that leads to an aberrant protein, a truncated protein. And so he wanted to come to the lab to work on this, and he actually wanted to come and use RNAi. And I said, well, I think we have something that's going to be maybe better than RNAi. And so he came, and he's now been in my lab almost 4 years. And so he's been really the driver of all of this, because he comes from a cell biology background. He had done a lot of work with mouse models before he came to my lab, something my lab had never done, never touched a mouse. And so we started thinking about how we might tackle this challenge of delivery, and especially delivery into the brain. It may be one of the hardest places to get targeted uptake of something. And so this is just a slide that summarizes different ways that we can deliver, many different people are using to deliver Cas9 and guide RNAs into cells or tissues. We can use plasmids that encode the protein and guide RNA. We can use viral vectors that are nice because they can have tropism. They go to particular cell types and not others. We can use messenger RNA that encodes the protein together with the guide RNA, and sometimes you can encapsulate that in lipid nanoparticles. And then we started thinking about this possibility over here, an approach where the molecules would not be genetically encoded, right? They would actually do what we do in the lab, which is purify the Cas9 protein, assemble it with guide RNA, and then somehow get cells to take up that assembled editing complex directly without having to encode it. And so that led to a series of experiments that Brett did, and then we eventually hired a gal named Jen Sabo, who's a mouse surgeon, basically, come in and start doing this in helping us do this in animals. But the idea was to assemble Cas9 with guide RNAs and then figure out how to get this to be taken up by cells, and either doing it in cell culture or, as I'll show you eventually, directly in mice. And so the strategy here was, and this was really a nice idea that Brett had, was to use a reporter cell line that had been created, and there's a sort of a reporter mouse line that's also available with the same reporter, in which in the genome is a gene encoding this red protein called TD tomato downstream of several stop codons. So naturally, under starting conditions, this gene, this TD tomato protein is not expressed. But if we target Cas9 to these sites, then we can actually edit out the stop codons and turn on expression of TD tomato. And if you do this in cultured cells in the lab, you could actually get results that looked like this, not this cartoon, but where you could actually see red cells. And so we thought this would be a great way to actually be able to screen for molecules that could get into cells and achieve this kind of editing. And I'm just going to summarize some of the data here very, very briefly. But first of all, so this is just showing that we could take the assembled protein RNA complex, called the RNP, and show that when you increase the amount that you were introducing into cells, and this is now just doing it, you're kind of cheating because you're using nucleofection just with cultured cells in the lab, you could show that the more you introduced into cells, the more editing occurred, and we could actually quantify this by separating the cells by FACS sorting. So that showed us that this reporter system was working. And then the question was, how do we actually get it into cells without cheating? And so, so the idea that Brett came up with was really sort of taking a leaf out of Carlos Barbas's book. So Carlos Barbas had worked originally on zinc finger nucleases and had the idea of putting cell-penetrating peptides onto those proteins. So Brett did the same thing with Cas9 and tested a whole series of protein constructs that had different numbers of these cell-penetrating peptides appended to the polypeptide sequence. And he found that he could then do an experiment like this, where he added this modified protein with a guide RNA to cells, and without nucleofection, if you didn't have any of the peptides on the protein, no editing occurred. But if we had several peptides on the protein, now we started to see red cells, and you could quantify that. And he found that the best results came about with a protein that had 4 of these cell-penetrating peptides appended to it. And furthermore, you could actually show, using a kind of a standard DNA editing assay here, that we got a lot of editing, shown by these shorter bands of DNA in this assay, with nucleofection. But if we didn't use nucleofection, we had to have the cell-penetrating peptide on the protein to observe these products that result from DNA editing. And you could show in biochemical assays that there was no problem having these peptides on Cas9 protein. These proteins behaved identically in DNA cutting experiments in the laboratory. And so then the question was, can we actually use this in a mouse? And so I'm just going to show you one example of the data that we started getting. So this is work done by Jen Sabo, who came and started doing stereotactic injections into the brains of these reporter mice, and then analyzing the resulting tissues. And it was quite a lot of work because she was able to not only look at the expression of TD tomato, which monitors editing, but also at the presence of different markers on the cell surface that would tell us what types of cells might actually be getting edited. And the point of this is just to show you that one of the surprising results that came out of this experiment was that while we found that there were numerous different types of neurons that were getting edited, as shown by comparing the presence of these markers with the TD tomato reporter, we saw no evidence for editing in astrocytes. And we think that's actually because, for whatever reason, these astrocytes are not taking up the Cas9 complex. And we're still trying to figure out why that might be and what's going on there. But it does sort of point to the possibility of eventually being able to do this in a designed way, where you could actually have tissue or even cell type specific uptake of these kinds of engineered complexes. And of course, we're excited to try to develop this further, and we've now started a very fruitful collaboration with a team of neurosurgeons at UCSF, which is our local medical school across the San Francisco Bay from Berkeley, to start doing this we have now 2 animal models of Huntington's disease that we're working in, and we hope to eventually work in a larger animal model and then look towards a pipeline towards starting a clinical trial eventually. And so I want to just end with a few thoughts about ethics. And, so again, for me, it's just been a crazy 5 years of, just sort of seeing the biochemical work that we started off doing now becoming a technology that's been adopted by many labs around the world and is being utilized to do all sorts of really interesting science, not only on the very applied side, but also lots of really fundamental, interesting research that's been enabled by having a tool that allows you to manipulate genomes in a targeted fashion. And as I mentioned before, I think the impacts across different fields of biology are going to be really exciting to see. So I thought I'd show you 2 fast examples of things that have come out recently that really caught my attention. One is this paper that Zack Whitman at Cold Spring Harbor Labs published over the summer, in which they were able to use the CRISPR technology to separate 2 alleles, 2 genes that are found in tomatoes that would be very hard to actually separate by traditional breeding. It would take a really long time. They were able to do this, and in the process made tomato plants that are much more robust in this sort of branching phenotype. So they can hold about 2 to 3 times more fruit than traditional types. And I'll tell you a secret. I went to Cold Spring Harbor Labs, and I talked to Zack, and I didn't want to tell him that I really wanted seeds from those tomatoes, but when I was leaving his office, he snuck me an envelope. So they're sprouting right now. So we'll see if I can replicate these results. I love tomatoes. So, and then I also want to point out that partly in honor of Mike Mellon, but I think there's an interesting possibility that these CRISPR systems could actually be used as antivirals. And I don't know about this particular strategy, but I think this paper caught my attention because it's sort of an interesting idea, and that is that you could actually excise integrated viral sequences from a genome, say a mouse genome, like in this paper, using the CRISPR technology. And thinking about how I think that might be a hard sell as a therapy, but I think as a research tool, it could be really quite interesting to see if you could manipulate cells and cut out different sections of viruses, et cetera. And then you may know that there already are clinical trials that are ongoing. This is kind of an older slide now from last year. But there now are 4 clinical trials underway in China using CRISPR in the immune system, sort of coupling gene editing with cancer immunotherapy, and more starting soon in the US. But what about this? What about human germline editing, something that's gotten a lot of attention? And Kathy Nyakun's work, published very recently, of course, is showing how this can be used very powerfully to get early information about early development in human embryos. But there's also work ongoing, published recently from the US, which has a more of I would say, a clinical focus. Really, the idea is, could we actually think about using this as a way to correct mutations in developing embryos. And of course, to me, the fundamental difference between somatic cell editing, where you're just treating one individual, versus germline editing, where you're creating changes that become part of the entire person, and not only that, are heritable to future generations, is really, now thinking about altering, if you really took it out far enough, altering human evolution, having a tool that allows us to do that, and all of the ethical implications of something like that. And so I've been very involved over the last several years in meetings to discuss this idea, leading to, at least in one case, this study that was published earlier this year by the National Academies in the US, the Royal Society also involved in this, and also the Chinese Academy of Sciences, to publish a sort of an in-depth study of this, and what are all the implications, and what are all the sort of ethical concerns, and to really try to come up with a path forward that encourages responsible use of gene editing. Where is this going? I don't know, but I think it's really an important time for scientists to be engaging in these kinds of discussions, not to dictate what should happen, but really to play one of the roles that we play, which is educating and encouraging active conversation. Because I think we've all seen this technology, and there are others too, that take off very, very quickly. And if people ask me what's my biggest fear is really just that we'll see a misuse of this that would cause some kind of a backlash against what I think is really an exciting opportunity to correct genetic disease mutations and do other things in science that I think we'd all like to see proceed as quickly as possible. So that's really my motivation. And I'd like to just close by acknowledging a fantastic group. Here's my group of my students that taken at a recent baseball game. Manuel, of course, is our original collaborator on the Cas9 research with her student, Christoph Czarlinski. These students I mentioned with Ahmet Yildiz, professor at Berkeley, doing the single molecule work. And then these folks are all involved in the applications of Cas9 to treat neurological disease. And of course, we couldn't do anything without money. And I always like to point out that I'm grateful to all of these groups, but in particular, the National Science Foundation actually gave me a very small grant back before anybody had heard of CRISPRs to fund one student. And that student was the person who really kicked off the whole project in my lab. And without that funding, it might have been very hard to do it. So I think this is just sort of a plug for small science, science that can really, in some cases, make a huge difference. Thanks.
So thank you for this wonderful, inspirational lecture. Now we have a little bit of time for questions. So we have two ambassadors that will help us in just you have to switch on the... Okay. No. Okay, so questions. There is one there.
So, just to go to the start of the talk, how does the bacterium survive being infected by the bacteriophage and sort of get to propagate the CRISPR?
Yeah, it's actually a really good question that's not fully understood yet. But one idea, so he's pointing out that when a phage infects a cell, if it's a lytic infection, there's not very much time before the cell is going to be dead. So how do I have time to do all of this? And so I think the best evidence right now in the field from a guy named Severinov at Rutgers is that it's the lucky cells that get infected by defective phage that have time to acquire sequences that then protect them from future infection. So that's probably what's going on.
More questions?
Jennifer, that was a beautiful talk. It's known that, at least I think it's known, that Cas9 and other Cas inhibitors can develop in the prokaryotic world. Is there any evidence that eukaryotic cells, you mentioned that the astrocytes were refractory. And you said that's because of lack of penetration of the complexes, but is there any evidence that any eukaryotic cells actually are resistant to the activities of these enzymes?
Well, did everybody hear that? Yeah, are eukaryotic cells resistant to Cas9 in some way? There's actually some interesting data that I saw recently from Matt Porteous at Stanford. So he's been looking at antibodies against Cas9. It turns out that because strep pyogenes is a pathogen for humans, a lot of people actually have detectable antibodies against Cas9. So that sort of raises an interesting question in the future about clinical use of this and whether we need to come up with other variants maybe that haven't sort of elicited that response. But beyond that, there's no evidence right now for inhibitors of Cas9. It seems to work robustly in systems that have been tested. It doesn't mean there aren't inhibitors out there, but they haven't been found so far.
Questions from the young people there, yes.
So you didn't talk about the off-target effects. So is there a relation? Is there a relationship between the seed sequence or the number of base pair mismatches and the off-target efficiency? So even in your base pair mismatch experiment, were those mismatches in the seed sequence or? Yeah,
so I didn't dive into the weeds too much on that. So you obviously know a lot about the system, but so very briefly, so he's asking, so if you think about the RNA-DNA hybrid that forms in Cas9, you've got 20 base pairs. There's an end that's closer to where the PAM is in the DNA. The base pairs right next to it are called the seed interaction, right? And that interaction is really critical for forming that unwound structure in the protein and leading to DNA cutting. So we actually couldn't do experiments to introduce mismatches on that end of the duplex because they would never bind in the first place. So all of our mismatches, the ones I showed you, are introduced on the other end, which is in gene editing experiments is the end where you tend to find, if you find off targets, that's where they're occurring. So we're really trying to figure out, is there a sensor on that end? And the answer is there is, right? And can you make it even better than it is in nature? So.
So now Cas9 actually used for the detecting RNA. So could you tell us how specifically, Cas9 can detect RNA transcripts and targets, or how can we actually use that for future therapy?
So there are definitely RNA targeting, sort of naturally RNA targeting CRISPR systems, and there's a recent report that you could use that sort of. Are you asking about that? Sort of use it sort of as an alternative to RNAi. I have to be very honest. I remain to be convinced about that interpretation of those data because we've been working a lot on those RNA targeting enzymes as well, and all of our data suggest that these enzymes are they're activated by binding to a target RNA, but once they're active, they don't work like Cas9. They actually work like general ribonucleases. They cut every RNA transcript that they encounter. And I just don't see right now how you could have one enzyme that has very close similarity to another that would behave fundamentally differently. So I don't know how to think about that yet, but I don't really think that these enzymes in the CRISPR system, at least for RNA targeting, are working like Cas9 to cut a single transcript, right? So. Hi.
Hello. So you've been at various institutions throughout your career, and how would you say that the atmosphere at Berkeley has played a role in the success of your research?
Thank you for that question. I didn't plant that question, by the way, but I'm very glad to get it because the answer is yes. And why is that? Well, I've been fortunate to work at a number of really great universities over my time in science so far, but Berkeley is interesting because it's a place that is kind of really, it's big, it's and a bit chaotic. There's a lot of people there, but it's universally, I've found it to be a highly collaborative environment. And I think that, you know, one of the keys, as I kind of alluded to when we started this work, was the fact that I got tipped off to this CRISPR system very early on. And that was because of someone who didn't even know me personally, right? But just, you know, was a scientist who was curious. She didn't do biochemistry in her own lab, but she realized there might be something interesting there. And she reached out. And that's been my experience over and over at Berkeley, you know, with people that we've worked with over the years. It's just an environment that's very conducive to collaboration. So if I have one piece of advice for students here, I think that, you know, especially right now in the biological sciences, to often, not always, but often to do the most cutting edge work really does require different kinds of expertise. So it really does rely on having great colleagues and collaborators to do science.
Last question, because then we invite you to talk to Jennifer maybe over a drink, or we like to offer something special to Jennifer before.
Thank you. That was such an amazing talk and the moment that we've been waiting for. Two questions, if I may, more on the translational aspect of gene editing. So you mentioned about, one is about the delivery, and in vivo gene editing versus ex vivo gene editing. So what is your sort of vision in terms of safety versus efficacy in these two different methods? And the second question is about, you know, who's going to pay for it? That's a great question. This is the epitome of personalized medicine. And one injection for $500,000, that's the sort of price we're talking about. So what is your vision on, you know, trying to reach out to the world on that scale, if that's okay?
Yeah. So regarding safety versus efficacy, I didn't mention this specifically, but it's one of the reasons we like the idea of a non-genetic way of delivering Cas9. Because I think many people's experiments, including our own, suggest that one way to have a safer gene editing tool is simply to limit the amount of time that the editor is, or that Cas9 in this case, is in the cells. And so what we find with these direct protein deliveries is that, you know, you deliver it as a pre-assembled complex, it's ready to go, and then within 24 hours, much of the protein has been degraded just by natural turnover. So it really does limit access of this enzyme to causing mischief by, you know, going towards off targets. But because it's pre-assembled in the cell, it actually gets going pretty fast as well. So you get pretty decent efficacy with, I think, a pretty good level of safety. We'll see if that proves to be true going forward with these animal models, but that's what we've seen so far in cultured cell systems. And then with respect to how do we pay for something like this? I mean, and I don't know the answer, but I've been interested I've been in conversations with a number of people from big pharmaceutical companies, and you might think that they would be resistant to this because, you know, they would say, well, you know, we don't want to have something that actually cures people. Hope I'm not offending anyone. But I think that what I've been encouraged by actually is that at least I've been talking to a couple of the big pharmas in the U.S. that are actually embracing this because they think that there's actually a path forward for thinking about how you could have a treatment like this that could in principle be a cure, priced at a level where, you know, you could, you know, make it viable for companies, but also thinking about a strategy for coming up with you know, you don't want to have to design new guide RNAs for every patient, right? That's just not reasonable. So thinking of ways of making sets of guide RNAs that would be able to treat a large number of patients that have the same genetic disorder. They might not have the identical mutation, but you would be able to perhaps excise a sequence, for example, and replace it. So, you know, I think it's an ongoing discussion. I think there's kind of a change going on right now in terms of thinking about the way that this alters at least some types of human therapeutics. And obviously there are some diseases where we really have no treatments right now at all, where this would be something that would offer a lot to patients if we could get it working. So, you know, it's an ongoing discussion, but I think it's one that's important to be having, not to just ignore it. Because I do think that, you know, there's a lot of clinical treatments that are coming forward right now that are exciting, but they're also incredibly expensive and so difficult for many people to get access to. And I don't really feel comfortable, working on something that's only going to benefit a tiny number of people. I really want to be working on something and working towards a day when something like this, if it comes to fruition and for clinical use, is widely available. It's not an easy question to answer, but I think it's one we have to be thinking about right now.
Thank you, Jennifer. We still clap for... And we have a special gift for you, Jennifer. It's a little heavy, but it's an engraved crystals with the DNA on it, which is given to all our guests with King's Cup.
It's really incredible. It's got the DNA double helix in the center, and I can see that you could have a light shining through it and see the. That's very nice. Thank you very much.